Vanithi's weekly podcast. Today will be a summary talk for future human circulating tumor cell therapeutics, liaising with my previous eukaryotic transcriptional proteins purification skills. Protein therapeutics play a significant role in medicine. The first 1922 therapeutic recombinant human insulin protein is 40 years old advancement. The micro and macromolecule proteins transport signals to the cells through receptors and membranes from one organ to other with intra and extracellular signaling and scaffolding support. Some of the protein therapeutics advantage over small molecules drug with their form and function are they are highly specific with complex functions that cannot be replaced by chemical synthesis. Less potential for adverse effects. Well tolerated with less likely to provoke an immune response. When the gene is deleted or mutated, protein therapeutics provide effective replacement treatment without the need of gene therapy. Interestingly, the FDA approval duration may be faster than for small molecules. Lastly, from a financial perspective, companies can obtain patents faster, which is attractive over small molecule drugs. Now, let's look at the benefits of stem circulating tumor cell spheres technology for future humanized protein production with an advancement and improvement to the field for less adverse events. Large-scale robust sphere culture system for manufacturing scale. You don't require human organ parts or big animal organs or E. coli for such novel therapies. In general, we humans tend to generate a human immune response against non-human origin medication. Cancer therapeutics were not humanized in the 1980s. Mostly it was murine. Later in the 1990s, human antibodies were introduced into clinical trials. Another example is Gaucher's disease. Patients were treated by the production of recombinant beta-glucocerebrosidase, changing from laborious human placental purification. In 2018 and the 2020s, the most popular and powerful tool, the Nobel Prize breakthroughs, CAR T cell therapies and CRISPR-Cas9 were introduced into clinical trials for treating various diseases. Its safety and efficacy has not been established but raised ethical concern in human embryo and sperm germline editing. Having said that, Britain was the first country to approve for improving fertility and miscarriages treatment for editing human embryos. With these fundamentals, the proposed augmented SFRP4 protein therapeutics will replace recombinant deficient or abnormal protein for cancer and other Wnt signaling diseases such as metabolic enzyme deficiencies, pulmonary and gastrointestinal tract disorders, immunodeficiency, and other human albumin disorders in the blood as circulating cells, stem cells, and cancer stem cells. Through extensive years of SFRP4 protein therapeutics experimental research evidence for various diseases, predominantly from our research group, I can confidently expect that the circulating tumor cell protein therapeutics will have an expanding role in medicine and healthcare for future SFRP4 liquid biopsy clinical trials. The US patented SFRP4 anti-angiogenic property on endothelial cells holds promise for ocular disease, age-related macular degeneration therapeutics like the pegaptenib sodium approved anti-angiogenic therapy. If anyone is interested in developing these Wnt signaling therapeutics for educational or commercial use projects, please contact us through the Medong Therapeutics contact page or Vanati at medongtherapeutics.org. Thank you. Stay tuned for next week's gene therapy, gene delivery, targeted therapies, wisdom words.